Hello everyone. What we're going to be talking about in this lecture is basically how that information that is stored in DNA, the genetic material, is executed in living organism, basically leading to the production of proteins. So the two processes that we'll be talking are the processes of transcription, where the information in the genes is translated or transferred to a temporary copy of RNA, and then the process of translation, where that information in the temporary copy in the form of RNA is finally translated into a protein sequence. So the different topics that we'll touch here. First of all, we'll go through some couple of experiments showing how this connection between the genetic material and proteins was actually discovered at the end of 1800s and in the first part of 1900s. Then we'll talk about the concept of the central dogma, which basically summarizes what I just said, how the flow of information passes from the DNA to proteins. Then we talk about transcription, with all the steps and so on, translation, all the steps, and finally we'll talk about how protein in the end can be also modified after translation is done, and then we'll finish up talking about how mutation can affect ultimately the structure of proteins. So, evidence of the connection between the genetic material and proteins. Well, the very first evidence was produced in the late 1800s by these two scientists, Garrett and Bateson, which were studying this disease called alkaptonuria, which is basically a disease we know now is genetic, but before then they didn't know that, where individuals are lacking an enzyme which basically breaks down this acid called homogentic acid. So they accumulate this acid basically in their body, and this shows in the urine. So when these people go to the bathroom and their urine is exposed to oxygen, actually the urine turns black. Now what these scientists observed, it was first of all that members of the same family in different generations would tend to have the same disease. So they noticed that this was actually a genetic disease. So whatever was carrying this genetic information from one generation to the other, they didn't know it was DNA yet, they knew that it was affected somehow. Not only that, but what they had observed is that this genetic disease was actually affecting the metabolism, was affecting the as we say, the breakdown of this acid. They didn't know that it was an enzyme, but they knew that this acid could not convert it anymore. And so what they did is they hypothesized that there was indeed an association between this genetic material, a change in this genetic material, whatever it was, and the metabolism. So the production of whatever was able to convert this homogentic acid into a byproduct. So that's the first evidence of association between genetic material, I should say more than DNA, and proteins. But the second big evidence was provided by these two scientists, Beadle and Tatum, which were doing their studies, in this case, using these fungus, as you say, which is called Neurospora crassa, very commonly used in the lab. These experiments were done in the first half of 1900s, in the 1940s. And so what they were studying was this fungus, which was known to be able to grow in a minimal medium. A minimal medium is basically a medium it's a collection of different nutrients which allow for the growth. Minimal means you really need very basic things, just a few amino acids and a few sugars and so on, and they will grow. So if you take some Neurospora crassa, you take the spores, the replicatory spores, and you put them in minimal medium, you will see that these actually fungus will grow, forming these colonies here, this white spot right here. So what they did, these two scientists, they actually took some of this fungus or fungi, in this case different colony, and they irradiated with X-rays. And what they observed is that at this point, by doing that, they were actually generating other colonies. Say you irradiated and you take this colony, transfer it to the minimal medium, that could not grow anymore in minimal medium. So some of these colonies could not grow anymore. They would die after irradiation with the X-rays. However, they could survive if the minimal medium was supplemented with some other nutrients, such as, for example, ornithine is this example, or citrulline. But these additional nutrients will be different depending on the colony you are studying. So they all had a need for different additional specific nutrients. So what they had generated is what they're called auxotrophs. So organisms that they need an increased additional nutrients in order to survive. So what they hypothesized based on what they were saying is that basically the X-rays were affecting some genes, okay, which were coding for an enzyme that was necessary to produce that specific nutrients that was lacking. So to overcome the lack of this enzyme, at this point, if you add the nutrients, this lack becomes irrelevant, right? That was their hypothesis. And so they tested the strains, and they determined for each one of them which nutrients was actually necessary. Not only that, but once they determined the different nutrients, for example, they determined that these would need ornithine, these would need citrulline, they determined the relationship between these nutrients. What is their relationship? Are they connected somehow? Is one synthesized from the other one? So not only they identified the defective genes, but also they built a pathway where these genes were involved. So just moving to the pictures is probably faster. So this will be the scenario that you get with a normal fungus. Okay, Neuraspora crassa, you put it in minimal medium. So even if you don't add anything, it will grow. In this case, it's not a plate, as I showed before. Those will be liquid, minimal medium. You will get it growing. So, and obviously, if you add any other additional nutrients, they will still grow. They don't really need them, clearly, because they can grow in their absence. But now, what they had isolated, for example, was some mutants called RG mutants that, in this case, though, they could not grow in minimal medium, right? However, they could grow if you added any of these other elements. If you added ornithine or citrulline or arginosuccinate, arginine, and so on. Then they identify another mutant, another strain there, which in this case, again, it wouldn't grow in minimal medium, but it wouldn't grow differently from RG. It wouldn't grow even if you add ornithine. But it would grow if you add any of the other three elements. And then they identified other mutants, which progressively were lacking more and more the capacity of producing any of this, right? So RG at this point would not grow in the presence of ornithine nor citrulline, but we would need arginosuccinase and so on. And RH instead wouldn't grow with any of these elements except for arginine. So what they realize is also that all of these elements, they are actually connected with each other in terms of synthesis. So citrulline is produced actually from ornithine and arginosuccinate from citrulline. So what they hypothesize is that basically these different strains were nothing else than mutants that had been affected by the X-rays through mutation in one of the enzymes involved in the synthesis of arginine through the production of all of these different molecules, which correspond to the different nutrients that you need to add to this different strain. So the RG mutants now would have a mutation on the first enzyme. So if you provide anything else after that, it would have the other enzymes and it could produce arginine. RGF instead would be missing the second enzyme. So even if you add ornithine or anything else before, it cannot really produce citrulline and therefore cannot produce arginine. But if you add citrulline at this point, it can actually reproduce. RG will be a mutant of the third enzyme. So if you add anything before that reaction synthesized by the enzyme 3, it will not grow. If you add something after that and so on. So basically what they did, they could figure out how X-rays will be mutating some enzymes, so some genes that will code for these enzymes, which then will lead to the lack of the capacity of producing some nutrients. Therefore, what they did, that show the first evidence that there is a relationship between genes and proteins present in living organisms. Moving on. What we start talking about now is the process of transcription and translation, which are part of what we call the central dogma. This is how any scientist would describe the flow of information from DNA, where this information is stored in genes, gets first transferred to a temporary RNA molecule called messenger RNA through the process of transcription, and then this messenger RNA is then finally translated through the process of translation into a protein at the ribosome. So we'll go one step at a time here. So this is just a description, we'll define them, but the two processes we'll talk about here when we talk to transcriptions, that's the process, the conversion of the information in the DNA, in the genes, into a temporary RNA copy, a complementary RNA copy. And then the translation is when this complementary copy gets finally, messenger RNA gets translated into the protein. Let's start with the process of transcription. So when we talk about transcription, we are talking about what happens in some specific sequences present in the genome called genes. Now you have coding and non-coding genes. Coding genes are those genes that give origin to proteins. Non-coding genes will give origin to other molecules like transfer RNA, ribosomal RNA. What I'll discuss here is about coding genes, how transcription takes place in coding genes. Now, these are sequences of DNA that are sparse in the chromosomes, whether it's a circular chromosome in a prokaryotes or a linear chromosome like the one you have. Now, these are short sequences that take about 1 to 2% of the whole genome. In your genome, 99 to 98% doesn't code for proteins. So only a small percentage there. And you have about 30,000, 20 to 30,000 genes present in the human genome. Now, these genes all have a specific structure consisting of 3 key parts, which altogether form what is called the transcription unit. So in each transcription unit, you would have one part, which is actually that sequence that ultimately, through going through some post-transcriptional modification, but ultimately is the one that contains the information to produce the protein. So that's referred to as the RNA coding sequence. Together with that, now there are also 2 regulatory sequences which basically tell where transcription should start, and that's the promoter, and where transcription should stop. Why do you need them? Well, exactly because these sequences take about 1 to 2% of the whole genome. You need to know where to go to copy this sequence into RNA. It would be such a waste of energy if you had to copy all your genome, even that 99-98% of your sequence that don't code for proteins, you had to copy them into a temporary RNA molecule. It would be such a waste of energy. By having regulatory sequences, you tell exactly the enzyme that has to copy these into RNA, which is RNA polymers. You tell exactly where to go, where to start, and where to stop. So this transcription process is basically again is where this RNA coding sequence gets copied into our RNA complementary RNA strand. This is done on the RNA coding sequence, as we say before, and only one of the 2 strands. So you can imagine how this molecule here, this gene, consists of 2 DNA strands, right, anti-parallel with each other, but only one of them gets copied. This is done by RNA polymers. Like DNA polymers, RNA polymers can only form RNA in the 5' to 3' direction. Therefore, it will be copying the opposite strand, the one in the 3' to 5' direction. Now, transcription is conveniently divided into 3 steps. You see the naming is the same as translation, but they're very different steps. So in the first step, you have what is called initiation. This corresponds to also the different parts of the transcription unit. Initiation takes place in the promoter, And basically what it does, it gets everything assembled together so that in the next step, in elongation, the RNA coding sequence can be copied into an RNA molecule. And then the third step, termination, takes place in the terminator sequence. Basically tells where to stop the copying of the gene. All of this is what's referred to as transcription. These will lead, at least in eukaryotes, to the production of a precursor RNA molecule, not to the final messenger RNA. So this temporary RNA molecule produced by this process in eukaryotes will have to undergo 3 additional changes, we'll talk about that, which are referred to as post-transcriptional modification, which are additional to the process of transcription. Let's start with the first step. Initiation is that process that takes place at the promoter level. Okay, and basically the point of this process is to get everything together so that the RNA coding sequence can be put together. So this involves the recruitment of the enzyme that is able to copy the RNA coding sequence, which is RNA polymerase II. This enzyme is recruited there through the help of other proteins referred to as transcription factors. These transcription factors recognize specific sequences of nucleotides in the promoter. So you may have a MyoD, for example, transcription factor that recognizes a 6 nucleotide specific sequence of DNA. Another transcription factors that recognize a smaller one or a larger one. But they all are attracted to the promoter by these short specific sequences. Once they bind there, then they help the recruitment of RNA polymerase II. Now, in prokaryotes, the key transcription factor is what is called the sigma factor, which actually binds to the RNA polymerase II before binding to the DNA. Now, these transcription factors are essential to determine when this gene has to be transcribed. So basically what they do, they turn on and off the transcription of this gene. So they tell the machinery, the RNA polymerase, when it's time to do it, when it's necessary to produce the protein corresponding to this RNA coding sequence, and when it's not necessary. Okay, there are many, for example, transcription factors that are expressed only early in the embryo development that determine the formation of the key tissues that then will form your body, and then they get turned off. Some of them are they turn on so many genes, and then they're not expressed anymore, and these genes get turned off. So they determine when and where exactly the gene is expressed. So it's transcription factors that allow for them. So for example, you have genes, again, stressing on where, you have genes that have to be expressed in your neurons, but not in your muscles, and vice versa. How does the gene know when to be expressed or not is dependent on the transcription factor expressed in the brain or in the neurons and in the muscles. So some of them are expressed only in muscle, not in neurons, and vice versa, turning on different genes. Now, all of these, as I said before, lead to the assembly of the RNA polymers at what is called the transcriptional start site. This is this +1, is the very first nucleotide that will get copied into RNA. The promoter is not copied into RNA. Only the RNA coding sequence here and the terminator, which will be removed, are copied into RNA originally. So this process of initiation leads to the assembly of the RNA polymerase here at the transcriptional start site, and second important thing to remember, allows for the opening of the 2 strands of DNA so that the RNA polymerase I can have access to the template that has to copy. This picture shows you exactly what I said. It's good to repeat things. So now you have the transcription factors here. In this case, this is referred to prokaryotes. You have this transcription factor, the sigma factor, which is bound to the RNA polymerase, and by doing that, it can take the RNA polymerase to a specific sequence in the promoter of a specific gene. So basically what is done, he has brought the RNA polymerase there, and he places it right at the transcriptional start site. here. And together with that, you have that this leads also to the opening up of the DNA DNA molecule here at the transcription start site, forming what is called a transcription bubble, which is something like about 12 to 14 to 15 base pairs big. Okay. So once this has happened, so once the transcription factors have recruited RNA polymerase in the promoter at the transcription start site, and they have led to the opening of the DNA forming the transcription bubble, initiation is done. The next step is elongation, and that's exactly where finally the RNA polymerase can copy the template strand in DNA, so only one of them, okay, into the precursor RNA molecule. RNA polymerase, as I said before, works in the 5' to 3' direction. So it can only copy the 3' to 5' DNA molecule into the RNA, so forming this temporary RNA molecule. Before that, though, the sigma factor, the transcription factor that brought RNA polymerase to the promoter, is released. So this signal to RNA polymerase 2 that it can start copying. RNA polymerase, unlike DNA polymerase, doesn't need any primer to start. It can start to produce the RNA on its own. It starts at the very first codon, called the transcriptional start site, the first nucleotide, sorry, not codon, nucleotide in the RNA coding sequence. And then when it does that, so RNA polymerase now is moving through the RNA coding sequence here. And when it does that, it's actually opening up the DNA ahead of itself and closing it behind. So what it leads basically, it leads to the transcription bubble basically moving through the RNA coding sequence. It looks like you have this bubble that is actually moving sideways. It's not really moving, but it's just opening ahead and closing behind. Okay. This keeps going. So RNA polymerase is reading the 3' to 5' DNA template, one nucleotide at a time, adding one ribonucleotide at a time in the 5' to 3' direction, complementary to the DNA strand. And this keeps going until it reaches a termination sequence. Now, there are different types of termination sequences. The one I'm going to focus is a very unique one, very interesting, very elegant the way it works. And but there are different ones that don't involve necessarily this structure here. They still involve release factors, or termination factors. But in this case, in this example here, you have some termination sequence like the one you see right here. If you look at this sequence, you will notice, so these 3' to 5' would actually be the template sequence that is copied by the RNA polymerase, right? So this is the one, GGG, TCG, that gets copied into CC and so on. So once this gets copied, this termination sequence, it will introduce in the RNA molecule two anti-parallel complementary sequences. If you read this sequence in this direction and this in this direction, you see they're actually complementary to each other. Where there is a C here, there is a G. Where there is a G, there is a C, and so on. What does it imply is that as soon as this RNA is formed, these two sequences will form a double helix. They'll bind to each other, and they will have a loop in between. This sequence here, or this structure here, is referred to a hairpin. Not only that, so this RNA molecule, as soon as RNA polymerase copies this DNA strand, forms this hairpin here. But then DNA polymerase also start copying this sequence immediately after this termination sequence, or part of termination sequence, which is very rich in thymine and adenine. So we introduce a lot of uracil and eventually adenine here. Why is this important? If you recall, the bonds between adenine and uracil, or adenine and thymine, but in this case adenine and uracil, are based on two hydrogen bonds, so are very weak bonds. So now you have that the combination of the formation of a hairpin sequence here in this RNA, together with the weak bond of this growing sequence here, causes basically the separation of the RNA molecule from the template, the 3' 5' template DNA sequence here. This causes also the release of the RNA polymerase and therefore causes the termination of the process of transcription. So this causes the release of the RNA molecule and the release of the RNA polymerase as a consequence of the formation of this hairpin and the weak bond between this uracil and adenine right after. Now, this will be the end in prokaryotes. Okay, so that molecule that is formed in prokaryote genes, it's ready to be translated into proteins. You see later on, it can actually be translated even while it's formed. In eukaryotes, there is a different story. In reality, many genes in eukaryotes in the RNA coding sequence, so that sequence that give origin to the protein, ultimately, that has the information for the proteins. In reality, that sequence that give origin to the protein is spread among other sequences referred to as introns. So you have sequences referred to as exons that actually do have the information to produce the proteins. But among them, there are sequences referred to as introns that do not contain that information organizing codons necessary to produce the proteins. So the RNA that is produced by the process of transcription, however, will include both. Okay, and is not ready to be translated yet. So it has to undergo this process of post-transcriptional modification, which consists actually of three different things. It consists of a modification in 5' and in 3', and the removal of these introns, as I mentioned before. So this sequence that do not code for the proteins. So now, first step, let's go through them. Modification of the 5'. Now, this modification is necessary for two reasons. The precursor RNA, so that temporary immature RNA molecule produced by transcription, is not very stable per se. It would actually be degraded very quickly if it was not modified. And so this is the point of doing modification in 5', adding a cap, and the 3'. The modification in 5' also allows for the recognition of the final RNA molecule by the ribosome as well. So allows this molecule to be recognized and bound by the ribosome later on through the process of translation. So what is this modification in 5'? It consists of the addition of a guanine cap, which is basically a modified nucleotide with 3-phosphate group, which is attached through an ester bond to the 5' here. So 3-phosphate and a guanine containing modified nucleotide attached to the 5'. And again, this protects from degradation, but also allows for this RNA molecule later on to be bound by the ribosomes. The second modification takes place on the 3', so at the end of the precursor RNA. Here you have some signal molecules here, which are referred to as polyadenylation signal and with a cleavage site. This basically tells some enzymes that basically this RNA has to be cut in this position, and you have to add there a sequence of 50 to 250 adenine nucleotides, which is referred to as a poly-A or polyadenine tail. So it gets cut here, and this long stretch of adenine gets added to that. Again, the function of this 3' modification is to prevent this messenger RNA ultimately to be degraded too fast once it gets into the cytoplasm. It increases the life. This final RNA, by the way, could last a few minutes, sometimes even a few seconds, all the way to hours. So it depends on the RNA itself. Finally, the third type of post-transcriptional modification happening only in eukaryotes, I stress that, is the removal of the introns, referred to as the intron splicings. As we said before, a gene might contain, not all of them do, some non-coding sequences, referred as introns, spread among the coding regions, the exons. These are the ones that would have the codons. We'll see what we mean by codons when we talk about translation later on. but the exons have the codons, the introns don't have it, so they have to be removed. However, when transcription happens, both the exon and the introns are copied, and so the introns have to be removed through the process of intron splicing, so that now the different exons can be joined together and you can have a continuous sequence of codons which can produce the protein. So how is this removed? Well, it's removed in two main ways. Some RNAs can actually splice themselves out. They don't need any help. They actually have self-enzymatic activity. Other RNAs, instead, they do that, what we call the spliceosomes. The spliceosomes are complex of SNRPs, or small ribonucleoproteins. This will be these SNRPs here. So it's a complex of RNA, small RNA molecules, and proteins combined together that have enzymatic activity. And so these, they form the spliceosomes, which is what will bind basically the two ends of the introns. So this end and this end will bring them together, and by doing that, it will join the end of one exon, in this case will be exon 1, to the beginning of the following exon, in this case exon 2. By doing that, now you have the two exons that get joined together, and the intron gets released as this circular molecule, which is referred to as the lariat. Again, some other RNA molecules can do it on their own without the help of the spliceosomes. Once these three modifications are done, now you would have the final molecule. Now, one thing I want to point to you that's very interesting is how this splicing process doesn't always happen in the same way for a specific RNA. It can happen in different ways referred to as alternative splicing. What do we mean by that? Well, think about one thing. There is one fact. You have up to 30,000 genes in your genome, but it's been shown that we can actually make 100 times proteins, more than 3 times more than the number of genes that we have. How is that possible? If for one gene you have one protein, that would not be possible. You would expect to have 30,000 genes. However, that is possible because some genes that do have introns, when they undergo the splicing process, in some cases or in some cell types, they get some introns removed, and in some other cases they have other introns removed. Or some exons get skipped in one form, in one type of splicing, and others in another one. This is referred to as the alternative splicing. Alpha-tropomyosin is an example. So this is, just going straight to this picture here. So this is the precursor mRNA of tropomyosin. So this is a protein that is essential for muscle contraction, and it's expressed both in your smooth muscle and your skeletal muscles. You see there are 3 types of muscle cells that you have in your body or in animal tissues. Smooth muscle cells are those muscle cells that are responsible for involuntary contractions of your, for example, around your blood vessels, causing blood vessel constrictions. Skeletal muscle cells are those cells that form fibers in your muscles attached to the skeletal, has the name skeletal muscle, which cause you to move, ultimately, to move the bones compared to each other, producing movement. So they're formed by different cell types. Tropomyosin, alpha-tropomyosin, is transcribed in both of these tissues, starting from the same gene, and produces the exact same precursor mRNA in both cells. However, when it comes to post-transcriptional modification in the smooth muscle, now the exons, here labeled as 1, 2, 3, 4, 5, 6, are joined together, skipping some of these exons. So exon 3 is skipped in smooth muscle, as is exon 10 and 11. These exons instead are different in the skeletal muscles, where instead you have that number 2 at this point. 3 is present, but number 2 is not there. Okay, and the same thing here. Number 12 is not present. Something that was present in the smooth muscle, but not in the skeletal muscle. By doing that, now you have 2 different forms of alpha-tropomyosin produced So different sequences ultimately of amino acid present in alpha tropomyosin is smooth muscle compared to the skeletal muscle. This thanks to the process of alternative splicings, where the exons are combined together in a different ways in different tissues. Now, what we talked about here, finishing up talking about transcription, and then we'll move to translation in the second part of the recording. What we talked about here right now is how the process of transcription takes place in coding genes. Okay, there are other type of genes which are referred to as non-protein coding genes that give origin to other molecules, non-proteins, so like transfer RNA or ribosomal RNA. And now these genes would have different type of promoters that work in a different way, and also the RNA polymers that are involved in these type of genes can be the same or different. In prokaryotes is exactly the same RNA polymers that transcribe both coding and non-coding genes, whereas in eukaryotes you have the RNA polymerase 2, is the one I was referring to earlier on, transcribes only protein coding genes, where RNA polymerase 3 transcribe transfer RNA genes and one of the 4 ribosomal RNAs, whereas the other 3 ribosomal RNAs are transcribed by RNA polymerase 1. So you have 3 type of polymers with different function in eukaryotes, whereas the same one in prokaryotes. So as a result of what I've discussed in this first part of the recording of the lecture, what you end up having in eukaryotes after the process of transcription and the 3 post-transcriptional modification, you will have what is called a mature RNA, which now is ready to be transferred first of all from the nucleus into the cytoplasm and then be bound by the ribosome to be read and translated into a protein following what is called the genetic code. I will talk about translation in the second part of the recording.